Are you a year 11 student that wants to get ahead of the game for their A-levels? Then you need to watch this video. Here is the first topic that you learn about in AQA A-level biology. Let's go. So it's called condensation and hydrolysis reactions. Hydrolysis reactions, anyone and their dog could deduce the fact that hydro means water and lysis means breaking down. So you use a molecule of water to break a bond, okay? So you need to know about glucose, and in particular the structural isomers of glucose. So you've got alpha glucose, in which the hydrogen is on the top, and you've got beta glucose, in which the OH is on the top. That's all it is. You just need to stare at it for like 5 minutes. Constantly draw it out, and then you'll memorize it for the rest of your life till the day that you die. Right, fair enough. You need to know about monosaccharides, disaccharides, and polysaccharides. Monosaccharides are the smallest molecular repeating units of a long chain molecule, which is a polymer. Okay, so basically monosaccharides, they're like glucose. They're like the shortest small molecules. Polysaccharides are long repeating chains of monosaccharides, and disaccharides is when two monosaccharides join by a condensation reaction. They don't actually have to be different. For example, you've got two molecules of alpha glucose which join by a condensation reaction to form maltose. Okay, which is a disaccharide. Another disaccharide that you need to know about is sucrose, which is formed from a condensation reaction between glucose and fructose. You don't need to know the structure of fructose, so don't fear. And then also you've got lactose, which is formed from a condensation reaction between glucose and galactose. So that's it. And then you need to know the test for non-reducing sugars. So you'll have learned from GCSE that you add Benedict's, you heat it in a water bath, and then you just test for the brick red precipitate, right? Now we extend our learning a bit because that only applies to reducing sugars like, I don't know, glucose. If you wanted to test for something like sucrose, which is, as I've just told you, it's glucose and fructose, then you actually need to break the glycosidic bond in between. And that brings me on to my next point. The bonds between glucose molecules are called glycosidic bonds, okay? So one hydrogen gets lost from one molecule and one OH gets lost from the other molecule. Condensation reaction, hence a molecule of water is released. Okay, what else do you need to know? So first and foremost, you add the food sample to dilute hydrochloric acid. You add dilute hydrochloric acid to the food sample and heat it in a water bath to the boil. Okay, then you neutralize it with sodium hydrogen carbonate. Then you add the Benedict's. Now essentially this whole HCl and sodium hydrogen bicarbonate, what happens is this allows neutralization to occur, which breaks the glycosidic bond. Okay, this means that then you can just carry out the Benedict's test as per usual, you know, everything you learned in GCSE, and then you test for the brick red precipitate. But you have to break that glycosidic bond in between. Right, fair enough. Another type of bond is a peptide bond. That's when two amino acids join together via a condensation reaction. And another one is an ester bond, essentially when one molecule of glycerol and a fatty acid join via a condensation reaction. And that, ladies and gentlemen, is all of the bonds and all of the condensation reactions and hydrolysis reactions that you need to know for the first topic of A-level biology. Thank you for watching.